Welcome to our Saturday's review session for the final exam. I wanted to go over a couple of specifics with you. You know, it's disturbing when you look out into the sea of beautiful faces and a student in the first row is reading a pamphlet about suicide thoughts right before the final exam. So I see that, sweetheart. And is that a class project that you're just checking for someone? Okay, because if you are having thoughts, you need to come and talk to me, please. Now I'm going to be like, oh, God.
I'm too self-centered to kill them.
All right, so that was frightening. I look up and you're reading that pamphlet and I'm like, oh, God. So let me try to calm your fears a little bit, okay? Okay, breathe. All right, enjoy your cookies and your cuties, but I want you guys to understand that your final exam, okay, and in all seriousness, your final exam is going to make a difference for students that are really borderline on their grades, okay? And that's the way my final exam is structured. It's going to count as the weight of 2 tests, so it does carry more weight because it's cumulative, and it's just like what you had first semester. You've been through this one time. You've done this before. You're going to be okay. Okay? You're just take a deep breath. Stressing and sign in. Stressing about it is not going to help your performance. The best thing that you can do is to prepare and to breathe. Okay? Because and to eat breakfast and get, don't stay up until 3 a.m. and study and do what you can. Please remember to use your interactive notebook as a great resource. Okay? Your interactive notebook should be a good resource for you because through your good note-taking and annotations of your notes, you've kind of pared down the most important topics. You also want to use this study guide and what we do here today to help you. The final exam is 105 questions, all multiple choice, and then there's a 25-question quest that goes along with just the body system. So we do need to get through all of this stuff with you, okay? Your textbook is a great resource, but I wouldn't spend countless hours reading through the textbook because that's going to give you so much information that you won't be able to see the forest for the trees. That's a saying, okay? You need to understand the main concepts, and if you're using just the textbook. Now, if for some reason, I don't know, your cat pees on your interactive notebook and you're no longer able to use it, which I've heard that story before, then if you are only left with your textbook, go back to the summary at the end and use the outline at the end of the chapter because that's going to break things down and make them more simplified for you as well. Okay, so I still have lots of things to add into your grades. Your grades are by no means finalized yet, and they won't be on the day of the test, of the final. Okay? I like to have time to be calm and grade. When I rush, that's when I make silly mistakes, and usually the mistakes are not in your favor. I enter a 5 instead of a 95 and things like that, So when you see your grades go down off of school loop, don't panic. That doesn't mean I'm done. That just means that I'm working behind the scenes. Okay? Yeah. When you turn in the final, probably not because it's 2-sided and I have to scan all of the first side and get those pushed off to school loop. So, I may scan the... I think if I grade the final in front of you, it gives you a false impression that that's what your grade's going to be in the class. And it's just one paver in the walkway to success. It's a new saying. I think I like that. One paving stone in the walkway to success. Yes?
So we won't see our grades until they're finalized after?
Exactly.
Oh my God.
You won't see your grades until they're finalized. And because... every time I put something into school loop, I'm hit with this barrage of emails. And I am compelled to answer emails. I do my best. And then I spend time answering all these emails that I should be grading papers. So I'm going to go dark and just get the grading done. and it's going to be like in the old-fashioned days. I know you guys don't have this experience, but we used to not know what our grades were until you got them in the mail.
Me too.
Yeah. So I know. We live in this world of immediate gratification. So I just want to let you guys know it's going to be okay. And I will. You do not get a cheat sheet for the final. You have the outline. So let's get to it. All right, we started talking about Mendel's laws yesterday. And I don't know if any of you noticed or if you get the notifications that the study guides that I have new videos posted to YouTube, if you have it set up to give you notifications. I did post videos yesterday that I recorded in second period. I posted those to YouTube. And I believe the second one that I posted, I think I'm going to title them by the chapter number so that you can go and watch just particular sections again if you feel that you have questions. So that way we can just break up the videos. So you should remember that Mendel studied pea plants. And the pea plants, a traditional example, it's easy to start with. When he looked out at his garden, he saw either tall plants or short plants. And he never saw anything in between. He never saw any mediums. So up until Gregor Mendel, they, of course, didn't know anything about genetics. They didn't know anything about DNA. But breeders that bred domesticated animals, they knew how to maximize positive traits in animals when they did selective breeding. And they would take, if they wanted a really strong horse, they would take their strongest male and breed it with their strongest female and ideally get stronger offspring. This is not the time to sleep, right? Okay. We don't want to sleep through this. We want to be awake. And taking notes and writing. Probably writing and typing into your outlines, okay? So up until Mendel's day, everything was considered, all these traits were considered to be blending together. But what Mendel saw, or didn't see, he didn't see evidence of blending in his garden. He never saw a medium pea plant. He only saw tall and short. And so he then crossed that tall plant with that short plant. And hopefully you guys all remember that he got all tall. And he thought, well, that's interesting. What happened to the short factor? So then he took two of these plants and he crossed them. And in this so this was what we call his F1. In his F2 generation, he got 75% of the time got tall, and 25% of the time he got short. Now, Mendel was an excellent mathematician. He didn't invent the Punnett square. That was someone else. But he worked with statistics and mathematics, and this is how, from a mathematical way, came up with all of his laws. So Mendel's first law of dominance, explains that one trait will conceal the other trait. One factor will conceal another factor. Okay. And an allele versus an allelic pair. An allele would be just a version of a trait. An allelic pair would be what is the genotype that you're actually carrying. If you're tall, if you're a pea plant and you're tall, you could be carrying two of the tall genes, or one tall gene and one recessive gene. Excuse me, sorry. So your allelic pair would be this. Your genotype is another way, I think, of saying your allelic pair. The law of segregation, to segregate means to separate. And so the law of segregation is when you have, I'm going to wipe this away. The law of segregation says that when you have a parent that is that, for example, that during the process of meiosis, the pairs of chromosomes separate or they segregate from each other. So you produce gametes that each only have one version of the parent. And so through the process of meiosis, each parent is haploided. It carries only one version of the trait. You should know how to definitely do a simple Punnett square. Remembering that the top and the sides represent the gametes of the parents. And then these squares represent the fertilized result, the zygote. So if you bring the boxes back together, any questions on Punnett squares here? Be very clear of the difference between genotype and phenotype. 75% of these offspring are tall. That is the phenotype. 25% are short. So if you say 75% tall, 25% short, you're talking about phenotype. Genotype is different. Genotype is 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. We have some other ways of describing homozygous and heterozygous. Here we can say homozygous dominant is the same thing as saying purebred. So, for example, purebred tall, you would know would be a homozygous dominant individual. What's the other way of saying a hybrid? What do we call a hybrid? Okay, hybrid is heterozygous. And so you would know that that was carrying one of each. So when we talk about animals, for example, and you talk about like a dog that has a pedigree dog, that's a purebred dog. It's never been hybridized with other breeds of dogs. So if you want a purebred Great Dane, then you know that it's come from 2 Great Dane parents who were pure, who had never had, that little terrier sneak into the backyard. Okay? So, and y'all know that with selective breeding, there's benefits, but there's also costs as well, because a lot of times when you're inbreeding so much, you're decreasing the genetic diversity of a species, and that breeds in good traits, but it also breeds in some negative traits. And a great example would be in dogs, how you have German Shepherds, for example, with such a high rate of hip dysplasia. Okay, where they require hip surgery, or they have to be put down and they don't live as long because of their hip problems, joint problems. Another type of dog it's notorious for having a genetic problem is the Dalmatian. So much inbreeding in Dalmatians that usually you can count on having at least one puppy in every Dalmatian litter that's deaf as a result of the inbreeding. And then you look at wild animal populations, and you look at something like cheetah. We have so encroached on the habitat of the cheetah, and then all of the illegal hunting and poaching of the cheetah, we've reduced the natural population of cheetah in the wild down to the point that there's such a small inbreeding population that, it's doubtful whether or not they really are going to be able to survive, even with all of our conservation efforts, because there's just not enough genetic diversity in the gene pool. And you should hopefully understand what I'm saying when I say gene pool and genetic diversity. It is all of our variation that makes us as a species strong. But I'm kind of getting ahead of myself because that's evolution, isn't it? All right. But I love to talk about evolution. Okay. Law of independent assortment. Let's look at that one next. Law of independent assortment. And in the law of independent assortment, we have to discuss two traits at the same time. And this is one of the topics that I think students tend to have more of an issue with. So we're going to stick simply with pea plants, okay? And we're going to look at the tall trait and also yellow pea pods versus green, okay? So yellow, this is going to be for green, and that's the recessive trait. Dominant is the yellow. Okay, and you know, T is for tall and little t is, big T for tall, little t for short. Okay, if this is the parent and it's a hybrid for both traits, when it forms its gametes, the pairs have to separate, the pairs of traits. So you're going to get new combinations of all of these traits. You could have a big T with a big Y. You could have a big T with the little y. You could have the little t with the big Y. And you could have the little t with the little y. This looks to me confusing. So I always think of like, I don't know why, it always reminds me of the FOIL method in multiplying polynomials. First, inside, outside, last. I just always, so I go the first one with the first one, the first with the second, second with the first, second with the second, and you're not going to go wrong. You're gonna get all of the combinations. So when you have an individual that is a hybrid for all the traits, there you see all of the different combinations of gametes that they can make. These are gametes. Let's count chromosomes, because this will help us remember our meiosis stuff. How many chromosomes are up here in the parent? Okay, good. I'm glad we're doing this. What this actually represents is, remember, chromosomes come in pairs. So there's the first pair, and then I'll make the other pair smaller. This chromosome carries the big T. This chromosome carries the little t. This chromosome here carries the big Y trait, and this chromosome carries the little Y trait. So how many chromosomes have I drawn?
2, 4. Oh, 4.
I have drawn 4, because there are 1, 2, 3, 4 chromosomes. So you have to have 2 alleles for every trait, because all of our chromosomes come in pairs. This is a real, I can see, again, you guys still feel fuzzy on this topic. Okay? How many different types of chromosomes are up there?
2.
We have 2 different chromosomes. This is one type of chromosome, and here is another type of chromosome. But there are 4 individual strands of DNA shown up there. Now, when this cell is going to go through meiosis, okay, you know what I'll do? I'll make them even different colors so that we can't even confuse them with each other. We'll go Christmas colors. There we go. So when the cell is going to go through meiosis, what's the first step? The first step is to copy your chromosomes. So I'm going to draw it in black, and there's the copy. Now remember, this chromosome was carrying the big T trait, this chromosome was carrying the recessive, this one was carrying the dominant, and this one was carrying the recessive. With the colors, it helps, doesn't it? Okay. So in meiosis 1, remember that interesting thing called crossing over happens, where these 4 chromosomes get together. So now I'm going to be challenged. I'm going to try to draw a tetrad. Okay, so there's my first one, and then it connected there, and then remember they each had a copy, so I got to change colors. See, there's its copy. So now all 4 of them are together, and that's the tetrad. And this forms during prophase 1. The significance of the formation of the tetrad is that you get chunks that can cross over between chromosomes. So let's say that this chromosome here, we're not talking about the T trait, we're talking about something else that happens to be carried along. It can swap between chromosomes. And so when they separate back out again, they can carry different chunks. So when we pull this back apart during anaphase 1, we can get something that looks like this. And you'll understand in a minute, why am I talking about meiosis when we were just talking about genetics? I'll explain why. So at the end of meiosis 1, you have 2 cells that form, and now they're haploid. They're no longer diploid because you've pulled the tetrad apart. But if crossing over has occurred, you get something that could look like this, where a tip of one chromosome gets exchanged with the tip of the other. And it's never going to be the exact same spots, so, oops, gotta reuse my eraser. And so that's why we get so much genetic variability. Because now in meiosis 2, these will separate again, and now you get 4 completely unique combinations. They switch over. Traits switch over between. So when I look at this, I know that this is a sister chromatid, and this is the other sister chromatid. This we call the homologous pair. Homologous pair of sister chromatids. So this is one sister chromatid, this is the other sister chromatid. And they're exact copies of each other. Now, where am I going with this with the law of independent assortment? Okay? To assort, if you have a box of chocolates and it says they're assorted, you have like a variety. And independent means that what one does doesn't determine what the other one does. So we have a variety of traits that separate out independently of each other. And so just because when crossing over occurs, you get on this chromosome the dominant trait, doesn't mean that on every other chromosome you're going to get the dominant trait. And that's what accounts for the variability that when you have a parent that is home this whole time, oh my God.